Hi everyone, my name is Jianfeng Ke. I'm a graduate student from University of Massachusetts Lowell. Today, I'd like to talk about expanding GTEx dataset with brain ontology-based graph neural networks to investigate genetic impacts on brain disease. In this study, we expand the gene expression data of 10 brain regions in the genotype tissue expression GTEx dataset to 103 brain regions, which expand our ability to learn how genetic variation impacts disease. For example, genetic variations can potentially lead to disease by influencing the downregulation of critical disease-related genes in specific brain regions that were not originally covered. The human brain, with its intricate network of diverse regions, plays a critical role in disease development. The GTEx program has been pivotal in providing transcriptomic and genotype data from over 300 postmortem donors. However, the GTEx dataset includes only 13 brain regions, missing key areas where disease often developed, and only 10% of subjects having all brain regions measured. In this study, we only include 10 out of 13 available brain regions. To address this, we turn to the Allen Human Brain Atlas dataset, which covers over 100 distinct brain subregions from 6 subjects, including crucial areas for disease research. Leveraging this, we developed a graph neural network model based on an expert ontology that predicts 103 subordinate or previously uncollected brain regions for GTEx subjects. Here is the architecture of our graph ontology model. Our model used region embeddings, gene embeddings, and the gene expression vector of 10 GTEx regions to predict the gene expression for 103 brain regions in the Allen dataset. For example, to predict the expression for region I of gene J for subject K, you just need to use the region embedding of region I, the gene embedding of gene J, and the original expression of the 10 GTEx regions as input. Then the model will give you the prediction. We trained our model on 6 Allen subjects and fine-tuned the model on 30 GTEx subjects with all 10 regions. We also developed extended graph ontology models to impute missing GTEx regions using a two-step process involving linear regression and our original graph ontology model. With that, we can also apply our model on those subjects with missing regions to predict the 103 brain regions in the Allen dataset. In the following slides, I'm going to show you some evaluation results of our model performance. In this plot, we show that our imputed expression closely matches real expressions. The two plots at the top display the gene expression of gene NGEF across 103 regions from 2 Allen subjects, while the two plots at bottom show the predicted gene expression for the same gene for 2 GTEx subjects. As you can see, their gene expression patterns are very similar. To better qualify the similarity, we use the DS score, a matrix previously employed to assess the consistency of a gene's tissue-specific expression across subjects. The definition of the DS score for each gene is shown below. In this plot, each dots represent a gene, and it shows the DS scores on the imputed data against that in the collected Allen data. The positive Spearman correlation here in the plot shows that genes with high DS score in the Allen dataset also tend to exhibit high DS scores in our predictions on GTEx dataset. We use figure 3C from the 2012 Allen paper here to show that genes were grouped into modules in the original paper, and they found that modules were frequently related to primary neural cell types and molecular functions. To investigate if our imputed gene expression can be used to reveal similar expression patterns, we also cluster the genes based on our imputation for each of the GTEx subjects and see if the clusters are similar to theirs. Here, we compare the adjusted rand index, ARI, of our cluster results with the cluster in the Allen paper using a 1,000 permutation test. The red dashed line is significantly greater than random ARIs, which suggests that our predicted gene expression retains expression patterns observed in the original Allen paper. We also evaluate our model performance by comparing our model with other models using Spearman correlations across genes. We compare our model with linear regression model as well as CP method and mice on 30 GTEx subjects with all 10 regions. We leave out one of the GTEx regions, cerebellar hemisphere here, and use other 9 regions to predict it. It's evident that our model outperforms other models on Spearman correlation. Similar results are observed in the comparison of our model with others across the remaining 9 regions, and also on the GTEx subject with missing regions, where the GEO model often achieves the highest Spearman correlation and occasionally obtains the second highest. In conclusion, by filling in the missing GTEx region for GTEx people, our method will allow us to impute brain-wide gene expression for 221 subjects with at least 5 regions, increasing the utility of the GTEx resource for EQTL discovery. Also, by predicting the Allen brain regions for the GTEx subjects, we will be able to link genetic to gene expression in previously uninvestigated regions, providing greater insight into how genetic impacts gene regulation to result in disease development. And here are the references. Thank you very much for your attention.